We are starting in biology your new chapter called the cell. As you know, a cell is a structural and functional unit of life. That is to say, it is the building block of which all living organisms are made, and the smallest unit of life capable of all the living functions. It is defined as a mass of protoplasm bounded by a plasma membrane. Cells vary in size. Most cells are very small, some may be very large, that is microscopic. They vary considerably in shape. Variation depends mainly upon the functions of cells. For example, a nerve cell has a cell body with long cell processes extending from it. Its function is to receive and transmit nervous signals. Some cells may exist as independent units of life. Such cells like euglena, amoeba can change their shape, but most cells have a fixed shape. The number of cells vary from organisms to organisms. An amoeba is single-celled, while a human being weighing about 60 kg may have as many as 60 into 10 to the power 15 cells. In unicellular organisms like amoeba, paramecium, or chlamydomonas, all the basic functions of a living beings are performed in one cell, while multicellular organisms have well developed division of labor. So, their different functions are performed by different organs. For example, we have a stomach to digest food, a heart to pump blood, and a brain to think. An English scientist, Robert Hooke, discovered the cell in the year 1665 while examining thin sections of cork under his simple microscope. He observed a mass of empty hexagonal chambers like a honeycomb and called them cells. Cell is the Latin word for a little room. Between 1838 and 39, Scaliden, a German botanist, and Scaven, a German zoologist, proved that plants and animals are cellular in character and founded the cell theory. They postulated that the cell is the basic unit of all life. Virchow, in 1855, proposed the idea that all cells arise from pre-existing cells. No cell can originate spontaneously, but comes into being only by the division of already existing cells. After the studies of plants and animal cell by D. Berry and Scholes, the protoplasm theory was established in 1861. According to this theory, the cells or units of plants and animals are tiny masses of protoplasm, each containing a nucleus. In 1884, Strasburger concluded that the nucleus is related to inheritance of characters. After the discovery of different techniques and electron microscope, our knowledge about the cell and its organelles has developed tremendously. Let us now look at different types of cell under a compound microscope. Let us cut a small part of an onion and separate a peel from one of its inner layer. Put this peel on a drop of water on a glass slide. Cover it with a cover slip and observe under low and high magnification of microscope. We can do the same thing by scraping the inside of our cheek with the flat end of a toothpick. Place this on a clean glass slide. Put a drop of methylene blue solution to stain the cell. Cover the drop with a cover slip and observe under a microscope. You will see some flat or folded cells scattered around. Oval dense structure in the center of each cell is the nucleus. The region between the nucleus and cell membrane is occupied by cytoplasm. Now, let us say something about the compound microscope. The majority of cells range between 3 and 30 microns. The detailed structure of a cell has been studied under compound microscope that use light and can magnify an object up to 2000 times, and electron microscope that uses beams of electrons and can magnify things as much as 500,000 times. Certain structures can be seen only under an electron microscope. These are called submicroscopic structures. The structure of a cell as seen under an electron microscope is called ultrastructure. Now, the microscope consists of, a compound microscope consists of a lens that is close to the specimen placed on the storage stage and is called objective. Usually there are two objectives capable of different degrees of magnification. The lens through which you see is called the eyepiece. The image produced by the objective acts as an object for the eyepiece, which magnified it further. Now, cell, tissues, microorganisms and so on that are viewed through a microscope are called specimens. A specimen is usually stained before being viewed. A stain is a dye which colors certain part of the specimen a particular color. This helps one to distinguish between different part of the specimen. For example, if you stain a specimen with iodine, any part of it which stores starch will show up distinctly because the starch will be stained blue-black. The stained specimen is placed in a liquid medium on a glass slide and a cover slip is placed over it. This process is called mounting, which helps to dispel air. Different mediums are used to preserve permanent slides which can be stored for a long time. Now comes the viewing through microscope. Suppose you want to work with the low power objective. First, you have to align it with the tube. When it is aligned, you will hear it click. Then, move the mirror until it directs light up the tube through the eyepiece into your eye. Next, place the slide at the center of the stage and turn the larger of the adjustment knobs until the objective is about a quarter of an inch away from the slide. Look through the eyepiece and keep turning the knob until the specimen comes into focus. Then turn the fine adjustment knob very slowly to get the clearest view of the specimen. If the objective produces an image that is magnified 10 times and the eyepiece magnifies this image another 10 times, you will see the specimen magnified to 10 into 10, that is 100 times its actual size. We can do an activity with the help of this microscope. Fold a fleshy leaf of an onion and pull out a thin peel with the help of a pair of forceps. Alternatively, break the leaf and pull the pieces apart. You will see a thin peel separating. Place a small piece, small enough to fit under a cover slip of the peel in a drop of water on a glass slide. Stain it with saffronin, then cover the peel gently with a cover slip. Make sure that there are no air bubbles. Observe under a microscope and you will see a very amazing magnified cell from that very onion peel. So, this was about the activity which we can carry out from a cell collected from an onion or our chick, etc., etc. Now, let us come into the main part, that is the structure of a cell. Structurally, the cell is formed of three parts: plasma membrane, cytoplasm, and nucleus. Plasma membrane or cell membrane. Let us discuss few lines about this very part. Each cell is bounded by an extremely delicate, thin, elastic, living membrane called the plasma membrane. It is about 10,000th of a millimeter in thickness. In plant cells, it is present on the inner side of the cell wall. The plasma membrane is made up of two layers of lipid molecules with protein molecules sandwiching it and embedded in it. The structure of plasma membrane can be observed under an electron microscope only. It is a selectively permeable membrane which allows the flow of only some substances into the cell and out of the cell. Thus, it helps in maintaining the distinct chemical composition of the cell. Besides protecting the internal structures, it also gives shape and rigidity to the cell. Viruses do not have any membrane. Dear students, this was about plasma membrane. Now let us finish up today with the functions of plasma membrane. Plasma membrane gives a definite shape to the cell. It provides protection to the internal content of the cell. It regulates entry and exit of substances in and out of the cell. It can internalize solid and liquid materials by infolding or extending around them. This is a process of active intake of materials. In animal cells, it is involved in addition, recognition, and in the formation of vesicles, cilia, flagella, microvilli, etc. This much is enough for today. We will continue our discussion in the next part. Till then, goodbye.